Figured that one out. Choose cell bio, got it. Okay. All right, it's on speaking up. Okay, so uh while this thing is what I'm still waiting for this thing to open our beautiful PowerPoints, I have some good news and some bad news. So I'll give you the bad news first, and hopefully the good news will compensate for the bad news. The bad news is that my flight was canceled for next week, and I really wanted to be in class by Friday. And unfortunately, the airline just decided to cancel my flight. And don't ask why. I have no idea. I'm fighting with these people forever, and it's not working. So I had to buy another flight, and I could not get one for next week. So I'll be going home by Saturday the 13th. So that means I will not be there for your first exam. But don't panic, because I do have office hours and I'll be available. And now I'll give you the good news. So since I will not be there physically for the first exam, I will allow you to have a choice to either take the exam online at the time when you have the exam in the class. Oh, I see people clapping, so I guess that's very welcome, good news. Or if you would like to come to class and take the exam, I'm sure it's not going to be like a lot of people because I really don't want to overwhelm Belle that probably just almost had a heart attack by now. So let me see a show of hands if you prefer to take the exam in person in the class. Okay, I had unanimous vote for online exam. No, okay, all right. So I guess how many people want to take the exam online? That was the question. All right, everybody wants to take the exam online. Okay. If you are scheduled to take the exam at the SAS office, you can do that, but I will also make the exam available for you online, and I'll give you the time that you are allowed to take the exam if you go to the SAS office. So you can take either at the office or online as you feel see more fit for your needs. All right. So everybody's cool with that? Yeah, yeah, okay. So now let's pick up on a few things that I had done here for this class. We will go here to our end of lecture 3. So I want to go over a few things. I have just updated some information on the PowerPoint itself. So I'm going to, upload the PowerPoint to the Canvas, right after the class, because I didn't have time to do this before the class. So I'm going to share my screen, which is going to be screen 2.
Not yet. I'll ask her after class.
Sorry?
For this class? Yeah. Oh, okay. Do you want to just ask her? Dr. Waziri, someone has a question here for you.
Okay.
Hi, can you hear me?
Yeah.
Can you hear me?
Yes, I can hear you.
Oh, okay. So I wanted to ask, last class, I believe you had uploaded a PowerPoint from I think 2024, and then you changed it to 2025 because I noticed on the 2025 lecture there was a lot more definition. So I wanted to ask if you were going to be doing that with today's presentation.
Yeah, I'm going to go over some things that I have changed and put some more explanation, because I think it's going to be more helpful to you guys. So I'm going to go over these changes that I had done on the PowerPoint. The topic is basically the same, but I think, every time I look back in my lectures, I'm trying to figure out what can I do to make it more intelligible? Because we go through this, or I go through this process that when I'm putting things in the PowerPoints, things are clear in my mind, but then if it doesn't come across clearly, then I mean, what's the use, right? So I'm always trying
Okay.
fine tune here and there to see how it can be better understood. Okay?
Okay.
All right, so here, coming back here to this lecture 3. Okay, so this is what we had last week.
Sorry. I said if you
Okay, so let's go here to let me see where I'm okay here.
Like the echo?
On this topic of okay, guys, and I'm going to mute the class because it's giving me some background noise. But if you have a question, please, Bell, click on the unmute button so then I can address the proper question. So, guys, actually, let me not unmute you because I really need the class to be, focused and silent so you can really pay attention to what's really important here because there's a lot of topics to cover. And as you know, the class can be very complex, but I really try to make it as easy as possible in as many bite-sized topics and points that I can have here. Okay, so here, when you go here to slide 5, that this is about the same as you have in your PowerPoint. You see that I have added a podcast here. So I'm going to bring here. Let me see, where is our class in the Canvas? Just a second, let me open here so I'll show you. In case you have not seen the podcasts, so let me bring them here so everybody sees the, do you see the modules here, the Canvas for the class? Yes, I see here in the background. All right, so here you go to the modules. Okay, so here's week 1. So here, okay, you find here your class video from last time. Right under the class video here, I put this lecture 3 podcasts. Okay, so when you open here the podcast, you're going to find 3 different podcasts for this lecture 3. So the first one really goes into a lot of these details on model organisms and how these ideas of using a system that is more that is less complex than a multicellular organism. How is it that we can use these basic principles to understand more about the more complex organisms and pathways? All right, so this is the first podcast that goes from slide 5 to slide 15. Okay, so after that, so here, when I go back here, so slide 5, so all of these that we covered before on the model organisms, I put in this very cool podcast. So until slide 15, okay. And last time we were going through the iClicker issues. Let me ask you, Belle, were you able to sign into iClicker?
Yes, I was.
Yes, and you started the class?
Yes.
All right, cool. Okay, so here, going back here to, oh, not the right one, this one here. Going back here to our Canvas, you see that you have another podcast that goes from slide 18 to slide 30. So these are the chemical components of life. Okay. And then after that, you have this third podcast that I really, really liked from slide 48 to 59. All right. So make sure you watch those podcasts because I found them really, really cool. And it took me a lot of time and effort to put this thing together. Okay. So I really want you to make the best of it because I find it's going to really help a lot. Okay. So that's the first thing on the podcasts. Another thing that I have added here, because I was having some questions from some of you related to these basic principles of chemistry. So basic chemical principles, atom type of behavior and atomic structure, atomic interaction and bonds. So we really don't have a lot of time to go into a lot of details on these very basic chemical principles. So this is why I'm adding the podcasts, and I have also added these Jove videos. So I arranged with the Journal of Visual Experiments, and this is an Elsevier platform. I arranged with them to allow free access for our class, okay? So everybody with the link can access these videos for free, okay? So here I put all of these very, short videos. They're like a minute and a half to 2 minutes long each. That's why there are so many of them, okay? So all this information on atomic structure, periodic table, and properties of periodic table, and also how to identify the elements, and then introduction to chemical bonds, and all these topics here that are already in the podcasts. Here you're going to find in the Jove video, okay? And what I'm planning to do with this playlist is to prepare practice quizzes for you. So the practice quiz, I think I can put it like right after this playlist. So it's going to be like at the bottom of it, but I'm still learning how to do that. And of course, those are not going to count for grade. So those are only going to be for your practice, okay? Because a lot of times, you have no idea, of course, what kind of questions I'm going to ask in the exams. And you're like, well, how can I practice more? We don't have like official quizzes in the class. So I'm going to put those practice quizzes there so you can be more familiar with the types of questions and how to study for the real exam. Okay. So these two things in the module, I just want to go over it. And let me see what else. Okay, so that's pretty much it. So you're going to see, as long as I have the time to prepare those, I'm going to put podcasts right after the lecture and right after the Zoom video. Okay, so everybody's cool with that? Yep. All right. So we went over this. So now let's get you the first points. Let's see if our iClicker is working properly. So question here, which of the following is considered the first model organism? Let me make sure I put this in a PowerPoint style so that the iClicker should recognize that screen. So Belle, let me know when you're ready to start the iClicker.
Okay, I'm ready to start it up.
All right, so this is a very easy question. It's considered here, which one is the first model organism? So let's start the question. Let's start the iClicker, and let's make sure that you get your points for showing up for class. You should have 30 seconds. Okay, let me know when the time is up.
All right, it's been 30 seconds.
All right, so everybody was able, hopefully, to get your answers in. And your answer here is? What's your answer?
They said E. coli.
Yeah, it is E. coli. All right. Okay. So this here, let me see, it is in the podcast. So I'm going to just jump through those. Just going to fly through them. Let me see here 1 second. Make sure that you go over these basic principles. Okay. I want to get here to hydrogen bonding. So absolutely, you need to have a very good idea of what is this hydrogen bonding business, what kind of bond is not a real bond, right? And how strong it is. It's, well, actually quite weak. So what is happening here? When you look at the bonds that keep molecules together, like the covalent bonds, you'll see that they really hold a lot of energy in the bond itself, right? Now, when you compare to some other kind of bonds that's not covalent, like for example, with ionic interactions, we see that when crystals are in nonpolar environment or just outside of water, these ionic interactions really are quite strong. Like if you look at table salt, for example, you see that there is a crystal that is kind of difficult to break. Now, when you drop the crystal in water, you know that you break up these ionic interactions, right? But when they are in nonpolar environment, you see that the ionic interactions are quite strong. Now, you look here strength of the hydrogen bond, and it's quite weak, and it's not a true bond. It's electrostatic interaction, okay? So how is it that you form hydrogen bond? You're going to form a hydrogen bond if you have a electronegative element close together with an electropositive element. Now, this electropositive hydrogen, right, it has to be covalently bound to an electronegative atom. So when you look at biological molecules, this electronegative atom is going to be either oxygen or nitrogen, right? So let's see here into more details what happens with this hydrogen bond. We know that is just electrostatic interaction, right? And not all of the hydrogen bonds are the same strength. You have some that are stronger, like for example, when you have a molecule that is holding the partially positive hydrogen, when you have a molecule that is aligning, look at this, aligning the electronegative atoms involved in these bonds, okay, the electronegative here, electronegative there, when these two are aligned with the hydrogen bond, then the strength of this hydrogen bond is stronger. You find that there is more electrostatic attraction when the partial negative charges of those atoms when those are aligned, okay? Now, when you look at this other situation in which you have a hydrogen bond that makes an angle with those partially negative electronegative compounds or atoms, then you see that this type of hydrogen bond is a bit weaker in comparison to that type of hydrogen bond. Okay, so not all of the hydrogen bonds are the same. Okay, so besides the hydrogen bonding interaction, you're also going to find this other form of interaction that is not as strong as hydrogen bonds, and it involves other molecules that not necessarily have this electronegative atom. Okay, so the other ones are called van der Waals interactions. And I had a very big confusion in my head, like what is really these van der Waals interactions? Like until a few years ago, and I finally had the light bulb over my head. It's like, oh, that's what it is. So I'll tell you what it is here in a second. And now you do have some other points, easy points to pick up. So the question is here. In the image below, another easy question. Molecules of water are represented by the ball and stick. Okay, so ball and stick model. And which of the following is represented by the dotted lines? And again, everybody should get maximum points with that. So we only have water here, right? Water different colors, making shaded, like distance and more close together. This is all water. So Belle, please, let's start that poll.
Okay, it's done.
All right, so what is your answer here? Hopefully everybody answered hydrogen bonds, right? Okay. Okay, so this is where we get to the cool stuff about water. So these slides, these images and videos I took from the TEDx talk by Dr. Pollack. And how many people have actually watched this video on easy water? Who knows about easy water in the class? Okay, show of hands, so nobody has any clue what I'm talking about. So this is gonna be really interesting. I always like to get, to talk to a group of students who never heard of easy water, right? Because when you go to a class like this and you're like, oh man, they're gonna talk about water again. And it's like this stuff I heard since I was in, I don't know, first grade. And it's like, do I have to waste my time with this? But then it gets really interesting because when you start looking at water and you really think that you know everything about water, then you get really surprised, right? Because here are some questions. First one is like, how do you know the water is really H2O like everywhere? How was this determined? But I'm not even going to get too much in that point. I'm going to say, um, how is it the water works in the first place? So when you just observe what is happening with a body of water, let's say that this thing here is in a beaker, okay, and then you have this dropper with just plain water, and you slowly drop this water, little droplets on the surface of this beaker water, you will see that the droplets don't immediately merge with the water, with the bigger body of water, right? I'm sure everybody has done this type of experience before. Like you go the drop, 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 and then you're expecting to see the water just merge, and then it doesn't. It kind of bounces around on the surface a little bit, like bounces a bit, and then it merges. You're like, how is that happening? Because if you do the same type of experiment using, let's say, a non-polar solvent, like if you use acetone, for example, you don't see these little droplets bouncing on the surface of the acetone. You see that they merge right away. So it's kind of strange. And you start thinking about it. It's like, that's odd. How is it that these little droplets are bouncing around before they merge? Are they liquid or are they not liquid? And then you start thinking, well, they kind of looked like little jello balls before they merge. So they didn't quite look like they were liquid. But then you're so used to water, then you don't even think much about it unless your job is really to study the water. Okay. And then you start doing like some other observations. You're like, okay, so what is going on here? We know that on the surface of the water, you have this surface tension, right? And what is holding this surface tension, it's really this property of cohesion. So water molecules that are holding on to each other, they have this cohesive property. So cohesion is when molecules of the same type kind of hold on together, hold on to each other. In comparison to adhesion, where you have molecules of different types that will interact. Okay, so cohesion is happening here. And because of this cohesive property of water, you get situations like this guy here, for example, that can walk on the surface of the water. Have you seen these little lizards go around like that? I mean, it's really cool, right? Does anybody know the name for that lizard? What's the name of the lizard? Oh,
Some
I can.
students said basilisk.
Huh?
Some students said basilisk or Jesus.
Yeah, it's the Jesus Christ lizard. I don't know the other formal name. Okay, well, walked on water. All right, so this is really cool. This is a very cool property of the water that has this huge cohesive uh, surface tension, right? And then you get to these interesting people who decided to do other experiments with water, saying that, well, water is the solvent of life, like all life forms on Earth and most likely on other planets too. It should be based on water, right? Or on Earth, at least it is based on water. So then what happened here? This experiment shows that if you put two glass beakers close to each other, and in one beaker, is not showing here, but you put an electrode, right? That is a cation. So here, your electrode, I think this is not there, so let's put an electrode there. Here's an electrode that is positively charged. And then on the other beaker, you put an electrode that is negatively charged. And then you connect both electrodes by a wire. And not only that, but let's say that you put some kind of charge. I forgot the symbol for a battery here. So let's see here. Okay, I'm not doing a good job with the drawing here. So let's see you have a battery here, okay? I'm not gonna try to draw here because this is okay. Battery. And you're going to run a current between these two electrodes, okay? Have you seen this type of experiment before? Do you have any idea what happens to the water when you run a current from one beaker to the other beaker? How many people have seen this experiment before? Let me see, show of hands. Okay, how many people are awake? Okay, good. So this is another cool experiment. Let me show you here what happens. I just tested this video and it really worked. I'm just gonna delete here the background and now look what happens. If you run a charge, look at this, look at this, look at this. The water communicates, not only communicates between the two beakers, but then you pull the beakers apart and you form this bridge of water that is not looking very liquidy. This thing is looking more like a gel of some type. Definitely not liquid water, or you wouldn't be holding this tension between the two beakers. And you can pull these beakers up to about 4 centimeters apart, and the bridge still holds. And then you're thinking, what is going on here? Right? This is supposed to be just water. And how is it that the water, first of all, communicated between the two beakers? And second, how is it that the water formed this bridge? Something is odd here, right? And now you start getting interested about the water. Oh, here's the Jesus Christ wizard running again. Okay. All right, so let's see what is happening here. So when you stop to think about it, you say, well, how is it that water, first of all, how is it the water is interacting with each other, the water molecules, right? So they do have some kind of social behavior that we don't quite understand. So here, like this cartoon of two water molecules socializing at a bar, and this guy is not very happy with this other guy. Anyway, the water has some social behaviors. And now we start thinking not only how water is socializing, but also how is it that is really moving around and interacting with each other and interacting with different molecules. Okay, how is this interaction taking place? So then these people running these experiments with the water, they noticed that if you have this interface of water, like for example, when you saw with the water tension with the lizard running on the water, this interface in that case was air and water. Something is happening that is changing the properties of the surface of the water, or the properties right at the interface, okay? So now they start thinking, is it only with air or is it with something else? They say, well, it should be also with something else that is hydrophilic. So they did this experiment is inserting in a beaker, so this is a beaker, they inserted a gel that could be like a polyacrylamide gel or some kind of even a jello, okay, so like more solid jello. So they inserted a gel in this beaker of water, and in the water itself, they just dissolved some little particles that that would allow them to see if water would behave in some kind of movement. So they wanted to see what would happen with these particles in case the hydrophilic gel was inserted in the water. So what happened is that they noticed that very close to this hydrophilic interface, right, the water, the water formed this empty space. So the particles were pushed away from this interface. And they said, oh, this is really cool. Look at that. You just stick something hydrophilic in the water, and now particles are pushed away. So you end up with a coat, right? A coating close to the hydrophilic thing that does not have these particles. So it's like a water blanket right there. And this is actually the same principle that we have with non-stick pans, right? Like the Teflon, Tefal, something non-stick pans. How is it that stuff doesn't get stuck when you're cooking something? Like you go to cook an egg, right, on the non-sticking frying pan, and then the egg doesn't really stick to the surface. This is that principle that this hydrophilic surface is creating an interface that repels molecules that are in the water. And you're thinking, oh, is there water in the egg? Of course there is. It's a cell, right? You have life form there. Everything is based in water, right? So this is a principle of how stuff doesn't stick to the pan, okay? And then it gets more interesting. If you shine light, any light, on that interface, look what happened with the distance between the particles and the hydrophilic surface. That distance gets bigger. So in order to give a name to this phenomenon here, they call this an exclusion zone. So this thing is the exclusion zone of water at the interface. And it was a certain size when it was just there, and then you shine light in it and the thing gets bigger. Exclusion zone gets larger. You're like, why light? What is light doing here? Right? Then you start thinking, let's run some more experiments and try to figure out what's happening here. So the first thing that they noticed was that right at this easy zone, right, the water molecules were behaving differently, and they were not just water. They were making these lattices that look like honeycombs, okay? So when they look at the molecular arrangement of these lattices, and here is showing only in 2D, but you can imagine this thing is really like in 3D, okay? So the arrangement of these lattices were actually changing the molecular structure of the water. And right at this easy zone, it's not H2O anymore. It's this thing that is more like H3O2 to allow for these lattices to form, okay? So now you're telling me that water is not H2O. Well, no, at the easy zone, is not. And it's not liquid. It's a gel-like substance. So now you go like, okay, so let's look at the phases of water. We know we have 3 phases of water, like 3 phases of most things. So we know we have solid phase. Okay, that's the ice. And then you have liquid and vapor. And this state of water is right in between the solid and the liquid. So this is a gel-like type of thing, okay? 4th phase of water. So the properties between solid and liquid, they're going to be a lot different when you compare that to liquid alone. And of course, it's not really solid, right? So it's just at the interface with this hydrophilic material. Now, when you look at what happens, how this easy water is created, and what are the things that are influencing the size and behavior, we see that when you look very close at the surface with the interface material, when you look close at the surface, you see that water tends to form this negative charge layer, okay? So that's going to be in the next slide here. All right, so right at the interface, you have this aggregation of negative charge, okay? So here is showing a droplet of water in the air. So the interface is between water and air, okay? Do you think air can carry electricity? Is it a hydrophilic type of environment, no, material? How many people think that air can carry electricity? Anybody took physics? No, everybody seen lightning? Hopefully, right? Yes, air can carry electricity. You have electrical charges in the air, right? So here, if you look at a water droplet at the interface between water and air, right outside of the droplet, you're going to find here a lot of negative charges, okay? So what happens here? Very close to the negative charge, you know that opposite charges attract, so you're going to find that there are concentrations of positive charges close to that negative charge of the interface, right? And now you start thinking about this and you say, okay, so let's see if that makes sense when you just look outside on a cloudy day and you see a lot of clouds that are kind of held together like a cotton ball kind of thing, right? And you start thinking, well, if water would not have this property of charge formation, then clouds would never form like that, like in a agglomerate structure. Okay. If water did not have these properties, then once it would evaporate, it would just disperse everywhere. Okay. But because of this easy zone, you have this separation of charges, and then something else happens because when you get two droplets of water kind of close together, the other droplet is also going to do this separation of charge. And now you're thinking, wait, but that doesn't make sense because if the droplets have negative charges, why would they get together? Well, because in between the negative charges, you're going to have a concentration of positive charges. And now it should start making sense because negative charges will attract. So the concentration of positive charges will bring together these negative charges of the easy zone. And what ends up happening is that the droplets of water will attract each other and they will make these structures or these clouds that really make all this water to accumulate in one site. Okay, so now you start saying that maybe this explanation makes sense when you look at this other phenomena we have seen before, that if you see these droplets of water bouncing around on the surface, it's because they do have this easy zone that is allowing this droplet to maintain its form and cohesive properties before it would merge to the bigger body of water. Okay, and when you look also at the surface tension of water, that would really make sense if you have this easy zone and not very liquid state of water, as well as in this situation when you have this bridge formation, that you clearly see this thing is not really liquid, right? It's more like a gel-like state, right? So what they call this is like colloidal crystals. Well, but colloidal crystals you get really when you have other things other than water that is also mixed in the water and not fully dissolved. Okay, so here it gets even like more interesting, right? So experiments with these electrical properties of the easy zone, as we mentioned, close to the interface of hydrophilic stuff, you're going to have accumulation of positive charges, and then a little bit further away, you're going to have accumulation of positive charges. And that should just sound like a battery, okay? So now you start asking yourself, like, is this knowledge useful? And how can you make use of these properties of water, right? We know that some places in the world don't really have electricity. So if you can devise some equipment that will have enough of this interface and easy water formation, you can actually generate electricity based on this charge separation. And of course, electricity can be used to light up a light bulb. Okay, so you can potentially get electricity from the easy zone of water, from easy water. Okay. Now, like with every battery, we know that the charges end up, being used up, and then the battery dies. And then you should be asking, how can I charge this battery again? Well, this is really easy because what actually charges the battery is light. In this case, you can have sunlight that can charge this battery, but not only sunlight, but most of electromagnetic waves in all the electromagnetic wave spectrum can actually charge this easy zone and get charge separation. Okay, so here we see this other experiment that was really cool, right? As I mentioned before, Here you have electrophilic material, and here you have water with those little particles. If you shine light on it, you're going to expand that easy zone. Okay, the exclusion zone expands. And now you're thinking, okay, you're getting a lot of charge separation, you're getting this gel-like material, and you're getting expansion of that. That means you're moving the water. Like the light is moving the water, is making the water move, okay? And then you're thinking, wait a minute. If I look inside of living organisms or inside of our bodies, right, we see that we have a lot of blood vessels that do make this interface possible. So you have there hydrophilic cells, right? The cells that make the blood vessels, they are hydrophilic. And you have this water-based substance that runs through our veins, right? And blood vessels. So you have blood that is water-based. And somebody had this idea, like, what happens if you put this type of hydrophilic material in a body of water? What happens if you shine light on it? Okay, so then they came up with this experiment here. So this is only a little piece of what could be seen in the microscope. So what they did here is that they got a beaker, glass beaker, with water and a little bit of particles to see if anything would happen, right? And then they drop inside the little beaker, they drop a little straw, regular straw. Oh, you can see the straw. I have a straw here, just a regular straw, okay? They put a little piece of a straw in that beaker with water, and they took it to the microscope. Well, maybe they had nothing better to do, no one knows. So they took it to the microscope. And what did they do? They turned the light on in the microscope, and this is what they saw. Look at this. The water started to flow, not only started to flow spontaneously inside of that straw, but also there was easy water, the easy zone formation close to the borders of the straw. Okay, the water was flowing and there was easy zone formation close to the border of the straw. So light itself created the easy zone and allowed the movement of water. Let's take a look at it again. Pay attention to that. Look, easy zone formation, you see here, easy zone formation and water movement. Those two went hand in hand, okay? So what this is telling us is that when you have hydrophilic material close to water, you're going to get easy zone and things are going to start moving around. And now you should even be asking these questions like, does your heart supply all the energy that you really need to move all your blood around? Or is the architecture of your blood vessels helping also with this movement of water-based substance like your blood, for example? And we know that this property by itself is not enough to push all the blood around, because, if your heart stops, you die kaput, right? That's it. But your heart is not working alone. It is working in conjunction with these properties of water, and is also getting energy from electromagnetic sources. It's like, what does that mean, right? Electromagnetic sources. And there comes here another bonus question for you, saying that, do you think electromagnetic waves can move water molecules? Well, everybody should get these freebies, like 3 points. You only need like 5 seconds to answer that question. So please, Belle, let's give them some free points. Let me know when time is up.
Okay, it's been 30 seconds.
All right, did everybody answer yes? Hopefully, yes. Okay. Another question here would be, do you use a microwave to heat up water? Well, that's electromagnetic wave, and it definitely heats up water, right? We do heat up the food in the microwave. So that's the same type of principle. Okay. And now we start thinking, how useful is this information when you start looking at cellular properties? Okay. So here is a very simple representation of a cell. Okay. And it's not showing the nucleus there, but you see that inside of the cell, you're going to find a lot of proteins, and the surface of each of those proteins are going to serve as this hydrophilic surface to create easy water. So we know that inside of the cell, inside of the cytoplasm, we have a lot of stuff that is organized there. And if you look at charge separations, you're going to see a lot of easy water formation. And then you look also at the surface, at the inner surface of the cell membrane, and we know that we have a certain potential that accumulates, that is negative charge, that accumulates close to the inner surface of the cell membrane, right? And then you look also at other molecules that are distributed, and here is not showing the nucleus, but then if you look inside the nucleus, well, since we don't have a nucleus there, let's kind of put a nucleus over here. Okay, so that's your nucleus, okay? And you look inside of the nucleus and you find these other co-molecules that are DNA molecules. And one thing that we know, DNA molecules have a lot of negative charges. So that should definitely interact with water in a certain way to allow the easy water to build up. Okay, so everything inside of the cell, inside of this living system, is based on easy water. Okay, and here, if you look at the structure of mitochondria, we see that there is a lot of hydrophilic surface within the membranes that allow for easy water formation. And we know the mitochondria is very much responsible for generating a lot of ATP, right? The energy currency of the cells. Okay. And so here comes this other question, right? Where do you get your energy, right? Well, we'll say from food. I don't know why there is a picture of a pie eating contest without their hands over here, but okay, from food, obviously. But besides that, you also get energy from light. So not just like sunlight, but magnetic and electromagnetic waves in general, in particular infrared light. Okay, so we know that infrared light is very important at building up easy water in our bodies. Okay, and where do you get infrared light? What are sources of infrared light? Anybody?
A couple people said the sun.
The sun, yeah, but I was looking for something that is not so shiny. Anybody use night vision goggles? Those detect infrared. Your own bodies are emitting infrared light, okay? Most things have infrared light, emit infrared light because atoms emit infrared light. Living creatures emit infrared light, okay? So besides the sun, your body has its own source of infrared light that is powering your own energy to make easy water. And that's like really cool stuff, right? So sunshine, and if you go in a sauna that, yeah, has a lot of heat, has infrared light that helps to build easy water. And they say, even like grounding, and that's like you guys are in South Florida, right? I mean, freezing São Paulo, right? But you go to the beach and you're walking, on the sand, barefoot on the sand, and that's called, grounding, and that can also help you to build easy water. Okay. So why is that helpful? Well, basically, it's giving you a further or a more explanation of what is happening to biological systems. because I don't even think that these topics were incorporated in modern textbooks, okay, up-to-date textbooks. But that's really what happening. There are lots of papers that are showing properties of easy water, especially when you get to control of gene expression and behavior of the DNA and RNA molecules. The easy water is very important for proper functioning of DNA. Okay. And we know that all biological processes are going to, they are dependent on water properties, right? So keep in mind here that you're going to have a lot of other interactions that are happening in water, like acid and base interactions, as you're going to see next, right? Solubility of charged compounds. So all chemical reactions in live cells are really depending on water, and our bodies are really made mostly of water. Okay. So that would bring us to the next topic of acids and bases. So this is, again, just a brief review. I will make another podcast with more explanations on acids and bases. Okay, so I'm just going to brush through the surface on this topic. Okay, so what is considered an acid and what is considered a base? An acid is a hydrogen donor, and a base is a hydrogen acceptor. Okay, so something that is either weak acid or a strong acid is going to donate a hydrogen that is going to allow the formation of a hydronium ion. Okay, so the hydronium ion is going to have positive charge, and then your conjugate base is going to have a negative charge. So acids and bases. Okay. Okay, so when you look at water, we know that water can serve as the weak acid, but it also can serve as a weak base. Okay, so here is the reaction that naturally happens in pure water. You're going to have your weak acid and your relatively weak base that is going to exchange hydrogen and make hydronium ion and hydroxide ion. Okay. So we know that this hydrogen ion is always being tossed back and forth between water molecules. Okay. And that's very important for chemistry. How is that hydrogen ion exchange happening? So we know that we have those hydrogen bonds, and look at the alignment of these two electronegative oxygens here, right? They are straight in a 180 degrees alignment, okay? So this bond is actually quite strong, this hydrogen bond over here. So this oxygen is pulling on that hydrogen because this is partially negative, that one is partially positive, and this is going to be pulling, and one point is going to snatch that hydrogen from the water molecule, and then you end up with the two ions. Okay. And that will bring us to this idea of how much do you have of these hydronium ions in a certain solution, right? How is it the same amount of hydronium and hydroxide, or do you have more hydronium ions and less hydroxide ions, depending on other conjugate weak acids or bases, right? So you have this pH scale, right? So if you have pure water, you're going to have the same amount of hydronium and hydroxide ions in the solution. I'm not going to go into details of how to get these numbers, right? But everybody should have had or simple inorganic chemistry and should definitely know how this works, right? So if you have a neutral solution, you have a pH of 7. pH is the negative log of the concentration of hydronium ions, okay? So you have a pH of 7 that is neutral. Now, if you have more hydrogen ions, you start to get acidic solution. Okay, so the acidic scale goes from 6 to 0. Okay, and if you have less hydrogen ions, you're going to start getting basic solutions. Okay, so basic goes from 7 to 14. If you don't remember how this equation works, make sure you review that. Okay, so with that, we get to this next topic, that is the properties of non-covalent attractions that are going to keep your molecules held together, right? Okay, so acids and bases, we briefly looked at that, and we have looked at covalent bond formation. So these non-covalent interactions really have to do with ionic interactions, hydrogen bonds, and the other types of weak interactions that are van der Waals interactions. And again, I did make the podcast for that, so I'm just going to fly through this and see if we can get to the end of this PowerPoint, because yeah, we are kind of behind. Okay. So here, non-covalent interactions, starting with ionic bond. This is when one atom transfer the electron to another atom. And this is when you have separation of charges. You're going to have one that is going to be positive and the other one that is going to be the negative ion. And if this is in non-polar environment, then you're going to have this ionic bond that is formed, and this is how you get your salt. Okay. Okay, so this is also electrostatic attraction. Is not a real bond, right? It has to do with changes in those properties. Okay, so let me see here. If you drop salt in polar liquids, then you're going to break up the salt. Okay, and water, yes, it is a polar liquid. So this is what happens when you drop salt in water. The water molecules are going to surround each one of those atoms, like dropping table salt, NaCl, right? So water molecules will orient their hydrogens that are partially positively charged to the negative ion. Okay, so this is your anion, and then orient the partially negatively charged oxygen to the positive ion. Okay, so there you get solution. And as we know, water is a solvent for all living systems. So when you drop something that is going to be surrounded by individual water molecules, you break up that particular compound. Okay. So here, the hydrophobic interactions. Please make sure that you listen to the podcast. They put it very nicely. They explain it really, really nicely, and it's easy to follow. Okay. So here, looking at interactions that are hydrophobic. Okay. So you have this, first, what we call hydrophobic interactions, that per se they are not quite hydrophobic interactions, but they're really going to happen when the hydrophobic substance is placed in the water. Okay, so it's really an indirect form of interaction that is caused by the water molecules that are going to orient themselves around this hydrophobic substance and push the hydrophobic substances to one place in order to have a better distribution of entropy. Okay, so entropy distribution here. We're going to talk more about entropy and enthalpy later. Okay, so this is this type of indirect hydrophobic interaction is really caused by water. But then you have these other interactions that are van der Waals interactions. Does this have to do really with the molecules themselves, that they're not really polar, but they can get partial polarity when they are in aqueous environment? Okay, so we have pictures for that. So here is what is happening. So if you have a molecule here, and this is not water, it's another molecule, okay? So what happens here, this is not hydrogen bonding, but is a molecule that would have a little bit of a partial negative charge on this side and a little bit of a partial positive side, positive charge on these other sides, but you don't have a dipole, okay? You have partial, like momentary dipoles that happen, but then this is very, very temporary, okay? It's not a constant dipole like a water molecule. So what happens is that if you have partial dipole, temporary dipole here and gets together with another molecule that has a temporary dipole over there. These temporary dipoles cause this momentary, this kind of very weak interactions among these molecules. So those interactions are really what you call these van der Waals interactions, okay? So this is happening with non-polar molecules, okay? So this dipole that you see here is not really a dipole. It's a partial exchange, partial shifting of electron clouds in non-polar molecules, okay? Okay, this actually is out of place because I talked about this before. All right, so let's see what happens here. If you have a non-polar molecule that is dumped in water, okay, and you have this interesting shift in this momentary dipole. So what is happening here? Here, the carbon would get a little bit of negative, a partial negative charge here, and that would attract the partially positive charge that accumulated on that hydrogen over there. So you're going to have this interaction that is momentary, but by itself is not strong, but when you put a lot of these interactions together, then it becomes significant. Okay, so this is all part of your, could be hydrophobic or van der Waals interactions. Okay, hydrophobic is really talking about very hydrophobic interactions that are pushed around with the water. Okay, it's the same type of principle between hydrophobic and van der Waals interactions. Okay, so this hydrophobic interaction is when the water molecule pushes the very hydrophobic molecules together, like oil, for example, okay? And then van der Waals is really the same principle. Okay, so now we start to understand what happens with interactions of molecules that are present in the cells, okay? So we know that they are going to have a lot of these little charge separations, momentary charge separations, and also possible complementarity with other molecules that they naturally would interact with, okay? So in case of enzymes, for example, you have a certain active site that has these different charges because enzymes are usually made of amino acids, and amino acids will carry some charge. So that active site is very specific to some other molecules that have like opposite distribution of charges on their surface. So you end up with this very specific type of interaction that is based on those electrostatic interactions. So momentary and accumulation of this electrostatic interaction will actually give a lot of specificity for the interaction of these two substances, of these two, in this case would be like two proteins, okay? Again, this is not covalent bonding, and it is momentary, meaning that things will get together and things will come apart. And you have this constant type of getting together and coming apart type of equilibrium reaction, okay? All right. So this is starting to explain what is happening at the molecular level when you see these proteins interacting with each other. Okay. So here is a summary of what is happening with molecular interactions. First, I have looked at the covalent bonds. This slide is slightly different than what you have, but I will upload this updated slide. Okay. So you have here your covalent bonds, your electrostatic interactions. And what are the electrostatic interactions? Well, van der Waals is also falling into here. So yeah, this should be over here. That ionic bonds and hydrogen bonds and van der Waals, they are all electrostatic interactions, as well as those weak hydrophobic interactions. These are all electrostatic, okay? So this one here is the only one that is not electrostatic. It's true covalent bond. Okay. Still, I think I have to clean up this slide here. Okay. And then we finally get to this side of biological molecules, okay? So we know that in order for cells to work, they are based on these biological molecules, okay? All right, so we know that some atoms are very common to show up in biological molecules, and we have seen before in the periodic table that you have these 4 atoms that are the most abundant in living systems. And of course, we focus on carbon, right? So carbon, hydrogen, nitrogen, and oxygen. And then you have some that are not very common, but they are very useful for coenzymes and also very useful for cell functioning. So we know the cells are made up of carbon. So carbon is really the most important after water. Carbon is really the most important element that makes up molecules in the cells. And this you should know really what is happening with the chemistry of carbon. So carbon has 4 missing electrons in the valence shell. Okay, so valence shell has 4 missing electrons. We know that this valence shell of carbon would be happy with 8 electrons. Then the carbon would behave like a noble gas, right? It would have stability. But if it has only 4, it's not really stable. So it wants to get 4 more electrons from somewhere. Actually, it wants to share 4 electrons from somewhere else, okay? It wants to make covalent bonds. Okay, it needs to make 4 covalent bonds in order to be stable, okay? So when it makes those covalent bonds, it's going to make those polymers, and those are the organic molecules that we have seen. So how is it that carbon is going to make 4 covalent bonds? Well, it can make single bonds and arrange them in different orientations. So for example, you can have chains of carbons, or you can have some branched type of conformation, right? So you see here that carbon is binding to 2 other carbons or 3 other carbons and making those branches. You can even have a 4th carbon branching over there. But yeah, it makes branches, and it can even make rings. Okay, so different orientations. Now, when you look at the basic building blocks of chains, you're going to find that you have this type of now the name of the CH2 just came out of my head, but you have building blocks of CH2 that are going to form the chains. If you have 2 hydrogen atoms bound to carbon, if you have 4 hydrogen atoms that are bound to carbon, you end up making a molecule of methane. And actually, this molecule is very, very stable. This is actually a gas, okay? And it's pretty much stable. Now, if you have 3 hydrogens and 1 bond that can bind to something else, so we call this a methyl group. Oh, ethylene is the name of the molecule. All right. So besides single bonds, carbons can make double bonds to each other, okay, or also to oxygen. So double bonds are possible. Now, when you look at the structure of these molecules that are made up that have double bonds, you see that they are not as flexible as the molecules that have only single bonds. They have a certain rigidity and planar structure in common, okay? And as you see later, these are called unsaturated bonds. And besides that, you can make those rings, right? They can have double bonds. And again, those structures are kind of flat, and they tend to be reactive. Okay. So now we're going to start looking at these chemical components of cells, and I will take like 5 more minutes before dismissing the class, and I'll tell you what's going to happen also with the rest of the slide. We're almost done. All right. So cells are formed based on these 4 different molecules. So we are going to have sugars and proteins and fats and nucleic acids. All right. So what is happening here? The building blocks of those polymers, right? You're going to have sugars, monomers of sugars. They're going to build up polysaccharides or bigger molecules that have the building blocks, right? Carbohydrates, polysaccharides. Then you're going to have fatty acids that are going to help to make lipids. You're going to have amino acids that are the building blocks of proteins. So those are the polymers, right? Then you're going to have your nucleotides that are going to build the polymers of nucleic acids. And I'm sure this is not the first time you're seeing this, but we're going to see really more details on how these processes take place. All right. So when you look at what is in a cell, we know that cells are really these little chemical factories. And if you look at everything that is composing the cell, we see that most of the things that are in the cell, making the cell, they're really proteins. Okay, so about 15% of everything that is in the cell is really made up of proteins. Then you're going to have a lot of things that are made up of RNA. So, I had to joke right now, sorry. So you have proteins, and you have RNA and DNA, but then if you put all DNA and RNA together, this is only about 7% of what is making up the protein. Besides that, you're going to have some polysaccharides. So this is a sugar part, right? So usually on the outside of the cell, you're going to have some polysaccharides. And then you look at everything else, and these are going to add up to about 6% or 8% total if you take out the protein and the nucleic acids. So most of the cell is really going to be composed of proteins. Okay. And of course, all of those are your organic molecules. Okay. And then when you look at all the functionings and the reactions of the cell, you're going to see that some reactions are really going to break down some compounds. So those are your catabolic reactions. And when these compounds are broken down, you're going to extract energy from the covalent bonds. Remember, covalent bonds can store energy. Non-covalent interactions do not store energy. Okay, those cannot store energy. Only covalent bonds can store energy, right? So when you break down the bigger molecules, you're going to get this catabolism side of metabolism, right? So you break down, you extract energy from the bonds, and you end up with your building blocks. Now, cells also have ability of using building blocks to build up the bigger molecules. So this is a process that uses up energy and is the process of anabolism. So building up things is anabolism. Breaking down things is catabolism. And if you put both of them together, you get your metabolism. Okay, so this is breaking down, this is building up, and everything together, all the sum of the reactions that are happening in the cell, that is your metabolism. Okay. Now, types of chemical reactions. Okay, I will be able to just finish these last 3 slides. Types of chemical reactions. You have reactions that give off energy when they make those bonds. So you have energetically favorable reactions, okay? At the end of the reactions, the products are going to have less energy than the reactants had. So those are called exothermic reactions. And you have other reactions in which you have to input energy for the reaction to happen. Those are energetically not favorable, and they are called endothermic reactions. Okay. So remember that when you're talking about energy, you're really talking about what is stored on those covalent bonds, okay? And all the other types of non-covalent bonds, they do not store energy. Okay, oh, and that's just a blank slide. So yeah, we did get to the end of lecture 3. Now, before I stop the share here, Okay, so we have only a few minutes, or you guys can run away. Well, one thing I'm going to tell you now. For the lecture 4, I don't like to run behind in the class, especially in this class. I am going to prepare more podcasts for lecture 4. Okay, so make sure that you watch those podcasts. And when we get together on Wednesday, I'm going to fly through the lecture 4 slides. And that's the time, basically at this time, I'm like basically flipping the classroom. Okay. So I hold you accountable to go through those podcasts. If you have questions, come with your questions. Okay. So you can ask those questions during the class. So I'm going to do a bit of a flip so that we can focus on catching up and covering everything that we actually have to. So if you have any questions or comments, I'll be hanging around here for some more time, just like as if I would be present in the class.
Online, it will be the same exact time, the class time, or no, it will be
I assume so. I've been getting a couple of questions for the exam, so I'll send out an announcement answering all the questions that I've been getting about it. So I assume it's going to be the same time though.
same time.
It's going to be in the same time as if you are present in the class.
Oh, yeah.
Yeah.
Okay, and also can I just do it in person in the test center over there? Yes. Hi, Professor.
Bye.
Hi, a question about the exam, the online exam. It should be the exact same as class time, or you want to open it from morning to evening?
No, it's going to be the same time as in the class, same time as class time.
Okay,
Mhm.
and then can we do it in the test center, just in person in paper, or not at home? Because by the time I have class before this class, by the time I get home, I will for sure I will just miss the exam.
If
That's why I'm
if you prefer to take it in paper format, you
yeah.
let us know. Actually, I'm going to put a poll in the modules for the exam, because I will talk to Bell, and we will offer the exam in the class in paper format. So you will have the option of doing that. But I
Oh
wanted
yeah.
to have a better idea of how many people will actually prefer to go to the class and take the exam in person.
Oh yes, thank you. Thank
Okay,
you.
you're welcome. So either, in the classroom or online, wherever you want to take it, but it's going to be at the time for the classroom exam.
Thank you.
You're welcome.
Bye.
Now that the last question, this time I couldn't answer it because he said that the server was reconnected.
Okay, what's your name?
Daniel Bermolina.
Daniel.
That's R-E-N.
S. Oh, sorry. S.
No, you're right.
Oh, R.
R-E-M-O-L-I-N-A.
R-E-M-O-L-I-N-A. N-A. Which question was that?
It was the last one, the one that says yes, and the one that you gave us.
Okay. Thank you. I'll check into that.
Thank you.
Mhm.
Have a good one. Thank you, Professor.
Hello, Professor.
Yeah,
Okay, you can hear me.
Yeah.
I had a question on, I think on slide 42. When it says like grounding negative charge builds EZ, what does that mean? Like the grounding? Can you explain that?
42. One second. Let me just go here to 42. Okay, so grounding is basically when you go barefoot and you step on the earth. Well, it can be dirt, it can be grass, or outside environment, sand by the beach. So that's called grounding.
Yeah,
So
but how does
your body has electrostatic charges, right? When you step on the what you call on the ground, there has a certain also electrostatic charge. The ground tends to pull your or work with your own electricity, with the electricity in your body. Okay, like when you work with electrical networks or circuits, you usually have a ground wire in order for your circuit to work better, for you not to get a short circuit in a way. Okay. Have you ever taken a physics 2 for electricity and magnetism.
Yeah.
Yeah, so
Yeah.
that's same principle, grounding electrode.
So to
So
make sure,
your feet will serve as the grounding electrodes, and you step, step in the on the earth and you're grounded.
I had a question. Is that similar to like the water where it's like it's negative, negative, and in between since it's positive, like it makes them both pull towards each other?
Yeah,
Okay.
yeah, and then you do have this interface between your feet and the ground, I would say.
Okay, okay. And I had another question. In my notes I wrote down ionic bonds form salt. Is it in a nonpolar environment or in a non-covalent?
This is non-polar environment. The ionic bond is a non-covalent bond. Okay, so when you have that in a non-polar environment, the ionic bond will form. So you have the crystal like salt crystal. When you dump it in a polar environment, like in water, then the ionic bond will dissolve. And then you don't have the crystal anymore. You're going to have the ions that form.
So in a polar environment, the ionic bond dissolves and the crystal doesn't form anymore?
Right. This is why when you put salt in the water, the salt disappears because the crystals dissolve.
And to make
If
sure the
you
ionic,
Yeah, if
oh,
you
sorry.
put if you put salt in oil, then that's not going to happen. You're going to have the salt right there. You're going to have the crystal in the oil. It's not going to dissolve because oil is non-polar. So the oil is going to allow that ionic bond to be maintained.
Okay, so in a non-polar environment, the ionic bond does form, correct? Just making sure.
In a polar environment, it will dissolve. You get the ions. In a
Yeah.
non-polar environment, you keep the crystal together. The ionic bond will be there.
Okay. Thank you. And to ask about the test, it would be like in the same like class time or like
Yeah.
all day?
No,
Okay,
same class time.
class time. Okay. And it would just be like online, like through lockdown browser?
Yeah,
Okay. How many questions would the exam be?
50.
50. Okay, okay. All
Yeah.
right. Thank you.
You're welcome.
Oh, really? Okay. Hi, Dr. Waziri. I have a question. So for the test, is it using a webcam or no webcam?
No, with the webcam.
Have a webcam?
Yeah.
Okay. Let's see what else. Um, Normally I take my test down at SAS. If I take it at home on a webcam, will I still have all my extra time added?
Yeah, yeah. With all the SAS, I will add the proper time that you have already set. So it's going to be the same time.
Okay. And then my last question is, because obviously we have a ton of slides to go through, is the study guide that you made like the best way to really channel all of the different slides that we have to study?
I would say the best thing is to understand what these slides are telling you, to know, to really get the principle, digested, okay? And be familiar with what is it that these slides are really telling you, okay? If you understand the material, then you don't really have a lot to memorize. You just really have to understand what's going on. And it really should make a lot of sense with the progression of the slides. So I'd say, the PowerPoints are really the summary of the chapters, and I'm really trying to add some practice quizzes, along the way. I'm going to be working on the weekend to put some of those up, so that you have more more practice. Basically, get more familiar with the kind of questions that I ask in the test.
Would you say that the test questions are slightly similar to what we've done in iClicker, or are they more challenging?
Mm, sometimes they are similar. Most of the times they are more challenging because iClicker questions are just like check in the class if everybody's awake, basically. Okay, but I will start putting more challenging questions to the no, for the iClicker as well.
Okay, and then my second question, I feel like I've asked this before, but just to make sure, I've had classes before where, like I just had one in the summer, where the test was literally if you read the textbook, you pass the test, but it's a huge textbook. So if I just use the PowerPoints, will I be fine?
Yes, because all of the test questions I take is straight from my PowerPoints.
Okay.
So what I
That's
emphasize in the
much
class,
better.
yeah, what I emphasize in the class, I like to ask things that I have talked about. So the textbook can be a bit overwhelming. It's just too much information. And once you go through the textbook, it's like everything seems important. So it's kind of difficult to really narrow down to what is it that I really need to know, since you also have other courses to take, and you also have to eat and sleep and have a life, so it gets to be, if you're going to sit down and study the text, you have to put a lot of time and effort. So this is why I really try to break down to the most important aspects of the chapters, really to make it more more digestible.
Okay, perfect. Thank you.
You're welcome.
Thank you, Dr. Waziri.
You're welcome. So I guess everybody is gone. So I'm gonna get out here of this Zoom and start the other Zoom. Thank you so
Wait,
much.
sorry.
Bye.
For the iClicker, is there anything that I have to save before I exit?
No, you just have to end the class and everything is automatically saved. Then I can go in the iClicker and select the correct answers if you didn't. But I see you probably selected the answers already, right?
Yes, yeah, I selected
Oh,
it as
okay.
we were.
Yeah,
Yeah,
so
someone
there,
mentioned it to me. I didn't know.
yeah, you just click on the X to close the class.
Just end class?
Yeah.
Okay, cool.
All right,
And
perfect.
then
And then make sure that you log out.
got
Yeah.
it.
All right, cool.
And then I just unplug the iClicker from the computer, right? It'll be fine.
Yeah.
Okay, awesome.
All right. Thank you so much, Bill.
Thank you. Have a good one.
Thanks, you too. Bye-bye.
Thanks.